This is the DocTrack Dental, Oral, and Craniofacial Tissue Engineering Consortium, a partnership between the NIDCR, the Michigan Pittsburgh Weiss Regenerative Medicine Resource Center, and C-Doctor Sister Resource Center. So collectively, we form the consortium, and today's the next installment of our Innovator Networking series, which will focus on commercializing academic technologies in the healthcare space. So we have two distinguished and very experienced speakers, not in the regenerative medicine space per se, but with a lot of experience forming companies and commercializing technology. Charles Hart is the director of the UCSF Catalyst program. Prior to coming to UCSF, he was at Threshold Pharmaceuticals and led the preclinical translational studies. He's also been the led the global clinical development program for hypoxia-targeted anti-cancer drugs. He had been a senior director of biology at Galileo Pharmaceuticals and director of drug discovery at Signature Biosciences. So a wealth of experience in the private sector before coming to UCSF to head their Catalyst program. Gary Hammer is the Millie Schimbeckler Professor of Adrenal Cancer at the University of Michigan. He's also the director of the Endocrine Oncology program in the Rogel Cancer Center at Michigan. He's had many roles in leadership in both the university and in private sectors, amongst them on the board of advisors for NCI, past president of the Endocrine Society, and most related to the talk today, he's the co-founder of two companies that spun out of technologies from his lab, Melendo and Sling Therapeutics. So without too much more of an introduction, looks like Charles will go first, and I'll hand the floor over to him, and we're excited to hear their perspectives, and there should be plenty of time for Q&A. So hand things over to Charles, and thank both Charles and Gary for agreeing to speak, and thank you for the audience for tuning in today.
Okay, great. Thank you very much, David. So what I'm going to talk about, I'm going to start off talking about translation, and then I'm going to use drug discovery as an example and talk about academia as part of the solution. And then I'm going to finish up talking about the UCSF Catalyst program that David already mentioned. So the concept of the bench to the bedside, the laboratory bench to the hospital bedside, and the intervening period of translation is obviously a key aspect of healthcare product development, from the fundamental discoveries, both at universities and in the private sector. And this translational stage can be very, very, very challenging. In fact, it's been referred to, I hate this term, but it's been referred to as the Valley of Death. And the FDA uses this term, the NIH uses this term. Actually, I got this cartoon, this cartoon out of Nature. And the concept is that these promising discoveries in the upper left at the laboratory bench never make it to the patient's bedside at the upper right. And why? One of the main reasons that this is the case is money. There's lots and lots of money for fundamental, basic, curiosity-driven research. If you look at the far left, UCSF happens to be the #1 recipient of NIH funds for a public university. I think #2 or #3 of all universities. And then at the far right, for those of you that watch television at night, the number of commercials, the number of yeah, the number of commercials focused on drugs is just unbelievable, and it just shows how much money the pharmaceutical industry has. And the thing is that the translational phase really is hurting for funding, and partly it's because a lot of the activities in translation, and again, just to point out, I spent 30 years as a translational biologist in the private sector. They're not considered creative or novel or clever, and so you can't get a grant to do them. And then a venture capitalist expects that they're already done before they fund your company. I'm talking about things like, carcinogenicity profiling, looking at, metabolism, and the like, translational types of questions. This is actually this graph. I took this out of this paper at the lower right, and I draw your attention to the fact that the y-axis, the left axis, is a long scale, and what you see is over the past 20 years, the number of papers published with translational medicine as a keyword has gone up, and I do have to point out, just to acknowledge, that term translation it's sort of a new term. I mean, it didn't really exist before 20 years ago, so I'm just pointing that out. But then on the right, I just put this together last night. You can't use translational medicine in the PubMed, but using the term translational research, you can see over the past 15 years how the number of papers has just exploded. And there's lots and lots of journals now focused on translation. Some of these are very prestigious, like the one at the upper left, Science Translational Medicine. The other thing I want to point out that's important, I think, is depending upon what type of healthcare product you're talking about, or we're talking about, the time and the amount of money, vary significantly. On the left, for a new drug, like 10 years and over $1 billion, and on the right for a digital health type, Fitbit, Apple Watch type solution or the like, can be a lot less time, a lot less money. And I already said this, I'm going to use drug discovery as an example today. Classic drug discovery, you go through a molecular targeted assay, and then a cell-based assay, animal-based assays, and human trials. And of course, there's phenotypic drug discovery, where you go right to the cell of interest, or even animal models of disease. And drug discovery follows this canonical, they refer to as a pipeline, from understanding the disease to identifying targets, and then identifying hits, advancing them to leads, then candidates in clinical trials. And there's this canonical pipeline. And it's funny, if you go into the Google image, this is a very, very, very common way to describe drug discovery. And then other people have argued, wait a minute, it's not linear. You really should be having a 2-dimensional or a 3-dimensional or 4-dimensional framework. But it is a useful, I think it's useful. And in fact, even though every single drug that's ever been discovered has followed a different path, I think the concept is, and then of course, once you get to the clinic, phase 1, phase 2, phase 3, and then approval, and some, post-marketing studies as well. Just to touch on the expense and the time, I draw your attention to the top, to the title bar and the subtitle. This data comes from 10 years, over 7,000 drug discovery programs, 1,000 companies. And if you look across from left to right, the success rate of transitioning from phase 1 to phase 2 is about 2/3. And so 1/3 of agents that start phase 1 don't make it to phase 2. I do want to say something that I think is also important. Companies, it doesn't matter if they're huge companies like Novartis or Glaxo or Merck, or a little 3-person company, they don't start a clinical trial unless they think it's going to work, because the cost is just so amazingly high. If you just look across this, I'm not going to go through this one by one, but at the far right, and this is important, at the far right, less than 10% of the drugs that start clinical trial get approved by the FDA. This slide here is the same data. It's a different paper. It's a different analysis. And the blue arrow points to what I just summarized, that across all indications, less than 10% of agents that start clinical study succeed. And if you look at the left, for some indications like infectious diseases, it's much higher, hematology much higher, and on the right, oncology much lower. I already mentioned this. Takes like 10 years to develop a drug. And this paper just came out. This is pretty thought-provoking, that if you look over the past 10 years, there hasn't been any improvement. Well, yeah, there hasn't been any, it's been taken 10 years for the last 10 years. This is actually a paper in Chemistry and Engineering News. This paper is 10 years old. And finally, this is the last slide. This is a little bit complicated because it's two graphs in one. The bar graph has the axis on the left, and these are the approvals by the FDA through the years. And if you see, it's pretty easy to see, you know, some years there's more, some years there's less. I've updated it in that box on the right, lower part of that box on the right. And so you see it jumps 30 and 50. 2 years ago, there was 50. Last year, there was 37. The point is that the FDA approves about the same number of drugs every year, year in and year out. The line graph is how much money is being spent on drug discovery and development, R&D, and it keeps going up. And so this has been referred to as a productivity gap. And I want to argue that academia is part of the solution. These are just mastheads of papers, you know, talking about the role that academia can play in drug discovery. And this is just a snapshot of resources for professors like Gary Hammer that's speaking next at University of Michigan, things that are available that they can tap into. I'm not going to read all these, but it's things like core facilities. I will describe the difference between a capital A accelerator and a lowercase a accelerator and the like. So these are some of the core facilities at UCSF. They do different things. And for the professors that are there, the price is usually at a great discount. If you're at a little biotech company in the region, you also usually get to take advantage of these. And I have myself at UCSF. And obviously you pay a lot more, but it's still fantastic. Incubators are these facilities that let a professor that has an idea that could be a company get lab space. And what's cool about these is you can just be like two people and getting one knee hole, and they're like, oh, that's completely fine. It used to be that you have to get like an entire lab. And in these incubators, you share the conference rooms and the bathrooms and the kitchens, you know, the photocopy machines, et cetera, et cetera. And these are all incubators right around San Francisco. Some of these are just totally unbelievably cool. The two at the top are sort of academic. The two at the bottom right are sort of real estate companies, you know, making money. And Lilly's and Johnson & Johnson's are corporate. Uppercase A accelerators. I know you've heard of these entities like Y Combinator, Techstars. If you have an idea, if a professor has an idea for a company, and you get accepted in one of these programs, they hold your hand and help you advance along translational path. And in return, they take a piece of the pie. I don't remember exactly what it is, but it's something like you get $250,000 and you give up 10% of your company. I should know. I think that's about right. And it varies. And if you go to their websites, and that's why I put this in here, they all have this these numbers underneath them. I'm not going to go through these. And I took these obviously right from their websites. So they all sort of claim the same type of success. You know, so many of our companies went public, they raised this much money. It's actually, you know, pretty cool. Lowercase a accelerators are like the Catalyst program. And maybe University of Michigan has one of these. Spark at Stanford is fantastic. They've come up with a playbook, and they've sort of cloned it. I think there's like 54 Sparks around the world. They do not take pieces of the pie. And so this is completely non-dilutive, and the funding the professors get doesn't result in them giving up a piece of the company. I threw this in just sort of for fun. There are these other programs that are, no, it's a crack up. They're called Igniters. So we got Catalyst. Anyway, this is sort of a new model. They're very, very analogous, but in addition to holding your hand, they actually do much, much more than that. And in return, they take a much, much bigger piece of the pie. Like, if you can believe it, up to 50%, but as low as like 25%. But the thing is, with these Igniter programs, they're basically doing the company, and in return for that, they get a big piece of they own a big percentage of the company. And of course, there are funding resources. One of the NIH's is NCATS. It's a center, it's not an institute, and then Osage. Yeah. Anyway, SBIRs, Osage partners, and this is just every college is different, but at UCSF, thanks to the Bayh-Dole Act, 45 years ago almost, the inventors at UCSF get a little over a third of all the patent proceeds that roll back to the university. Okay, UCSF Catalyst program. We're part of the Tech Transfer Office at UCSF. There's 5 other programs within this. I'm not going to read through all these. I'm going to talk about the Catalyst program, but one's the industry partnerships for either companies that want to interact with UCSF professors, or if a UCSF professor has an idea that he or she thinks a company might be interested in, they facilitate that. And the top left is our classic Tech Transfer Office that deals with IP and licenses. And okay, Catalyst program. We support therapeutics, diagnostics, medical devices, professors that have an idea that could be a product. And if you look in the text at the bottom, the key to our program is we have over 100 volunteer industry expert advisors. And these tend to be people that are older, like me. You're not supposed to say older, late career is what you're supposed to say. These tend to be late career people that have the time to give back. They're not coaching soccer or grading, helping their kids with homework. And we also have seed funding. We also have this really active internship program. Last summer we had 80 interns in the summer. I'll mention that in a second. The types of products, the types of healthcare products we support, very broad across those 4 types of products. We also support tools and platforms, and then there's, of course, there's hybrid products. There's medical devices with digital sensors. There's software as a medical device, etc., etc. And again, not just therapeutics have a pipeline, but diagnostics do, medical devices do, digital health solutions for patient benefit do. This is a snapshot of the expertise of our advisors. I'm not going to go through these. I'm just going to highlight a couple, like preclinical tox, regulatory affairs, prototype development. It's not surprising. It's not surprising that professors, wherever they are, aren't experts in this, because they're worried about what they're worried about. And so basically the Catalyst program helps professors with ideas, advance them, and then exit to whether it's an incubator or an SBIR grant or an igniter or what have you. This was last year's snapshot. We got 54 applications. Those got narrowed down to 19 finalists, and we gave 15 awards. And I just draw your attention to the very, very bottom, that after the awards, the thing doesn't end. It's not like, oh, here's your award, good luck. But there's a lot of hand-holding after the awards, and the awardees put together target product profiles, and they take I-Corps training. These are just the 15 awardees last year. I was going to draw your attention to these 4, but I just looked at my clock, so I'm just going to move forward. Really interesting projects. Catalyst has been going since 2010. We've awarded 279 projects. They've gotten over $10 million, and these same projects have raised over $500 million. And these are some of the startup companies that have either been founded based on Catalyst projects or in license Catalyst projects. And the 4 I circle with the oval are just recent from last year and the year before. These are sort of obvious things that would be important to prioritize a project for funding. And this is sort of like what they go through. You know, I can send these slides to whoever. Yeah, I know I'm going through this fast. I can send these slides to whoever's interested. But to those of you that don't know about I-Corps, it's fascinating. It stands for, the NSF started it. It's based on the model called the Lean Launchpad. Steve Blanket, a business professor at Stanford, came up with this Lean Launchpad model for company formation, and NSF really got into it. This is the regional I-Corps between UC Berkeley, UCSF, and Stanford University. Basically, if you look, I did this last night too. The top of this slide on the right, that small text, that's the first 10 pages of Ozempic's product label, the package insert, the prescribing information. And basically, a target product profile is a prophetic view of what the sponsor wants that to look like 10 years out. These are the fields that go into it. This is our little network. And anyway, thank you all very, very much. I'm going to turn it over to Gary Hammer.
Gary, you're muted.
Hey, Gary, you're muted.
We good? Can you hear me now?
We can hear you now. Thanks.
Great. Charles, that was outstanding. My task, everybody, is to give a small personal story that illustrates some of the essential points raised in Charles' talk. So my talk's about 21 minutes long. We'll have plenty of time for Q&A in a few minutes. So as Dave already mentioned, I'm part of the Michigan team here at the University of Michigan. And I've been involved in just a couple of companies, advising for some, but founding a couple. And I'm going to tell a story today about Sling Therapeutics, because I think it does a better job at illustrating some of the key issues that Charles and I are trying to get across about industry academic partnerships. So we're going to jump right in. Most of you probably know the Nobel Prize in 2023 given to Drew Weissman and Catalin Carrico for their mRNA work that got the COVID vaccines going. Some of you may know the Nobel Prize in 1950 given to Kendall Reichstein Hench for the discovery of adrenal steroid hormones, which is my area of interest. I suspect that only a few of you, however, know about the Nobel Prize in 1948 given to Paul Muller. Okay, Paul Muller was given the Nobel Prize for the discovery of the pesticide DDT. Okay, DDT was sprayed on everything, our food, our children, our clothes, to take care of insect-borne illnesses, as you probably know. Now, what's interesting is that in 1948, some of the early pathophysiologic tox studies done showed that DDT and its metabolite were toxic to dogs. And specifically, they were toxic to the dog's adrenal gland and caused adrenal failure. Okay, so Lesson 1, thinking outside the box. Some smart investigators hoped to and did turn this toxicity into a therapeutic asset. Okay. And in 1970, after 20 years of work, DDD, the metabolite, was shown to be effective in treating adrenal cancer. And it was the first ever approved drug for this disease, approved by the FDA in 1970, excuse me. At the same time, however, you might know that despite that FDA approval, DDT single-handedly ignited the environmental movement, founded the EPA, basically, and the EPA banned DDT from the earth in 1972. Now, the problem is, despite that, DDD remains the only approved drug for adrenal cancer in 2023. That's a problem, okay? Difficult to dose, extremely toxic, and it works in like 10% of patients. I'm a clinician, I take care of these patients. That's what I've got to work with. So that's the problem. Adrenal cancer is an extremely rare disease, maybe 1,000 cases per year in the US. That's a whole nother discussion about rare diseases. But the problem with rare diseases is there's no understanding of the disease. There's no standard of care often. There's no incentive to study. Why? The return on investment for pharma is too small. The return on investment in terms of lives saved for governmental agencies is too small. So we have a big problem. Moreover, most doctors who see a case of this disease have never seen a case before. They don't know what to do, and we have no guidelines. So we're in the hole to begin with. So lesson 2, leverage. So how do you leverage? What do you leverage to make a difference? Okay, it's about the power of team, not or team. So Michigan's been studying the adrenal for a long time, 7 decades. But, you know, things come and go at universities. Professors come, they go, they get old. But I was hired in 1999 to try to grow an adrenal program. And this slide is 20 years in the making. So, but we've been able to do that at Michigan with folks doing research, folks doing clinical care. We're embedded within an endocrine oncology program that takes care of patients with endocrine cancers like adrenal cancer. And what we've learned is, as many of you know, to make a difference, not only as a team, it's about collaboration. And we've developed 2 large platforms of engagement around the world with over 100 universities, 5 continents, where we leverage ourselves together to understand the disease and get grant funding and turn those therapies that we ultimately develop into clinical products. But it takes a big network to do that. So we've worked hard to understand the disease, develop new therapies. And of course, this is an iterative process, as you just heard from Charles. You know, what we learn from our basic science studies, my emoji of the mouse, inform us about questions to look at with patients. What we learn from our patients informs us what studies to do in various model systems. Iterative process. Now, when I started at Michigan, I was schizophrenic. On one level, I'm looking this way and doing basic science, and then I'm looking this way and seeing patients, but the two never met. I was 2 separate people. But slowly over the years, this has come together with the help of the team. And I'm going to tell that story and about how a company was founded based on that intersection. But as you just heard from Charles, this is a tough lift. It's a tough lift, and you just heard about the horrible term Valley of Death, where a lot of basic science has routinely been done at universities, translation done in industry, and the Valley of Death is a tough place to cross and merge. So I'm going to start with a little video for you. This will keep everyone awake before they have their lunch. Valley of Death.
But Dad, you'll never make it.
Don't you think I know that? Goodbye, son. Oh, thank God, thank God, thank God.
Dad,
I love you too, son. You know, boy, I don't think I've ever felt as close to you as I do right now. I'm gonna make it. I'm gonna make it. This is the greatest thrill of my life. I'm king of the world.
All right. Well, there's the problem. How do we avoid the Valley of Death and the stereotype of Homer Simpson? Well, the answer is discovery is iterative. That's the first example. right? Takes teams, basic and clinical, right? Across a variety of platforms that we just kind of heard about. So that's what Sling's about. I'm going to talk about Sling Therapeutics. So we've known for a long time in the clinical space that there are 3 familial syndromes of cancer with germline mutations in which adrenal cancer occurs. Colon cancer, Wnt signaling. Beckwith-Wiedemann syndrome, IGF-2 overexpression. Lee-Fermini, loss of cell cycle control. These 3 pathways are critical for these syndromes, and adrenal cancer happens. So we kind of already had a hunch that these 3 pathways were involved in adrenal cancer. Okay, we're going to focus on IGF. So we early on made a mouse to try to look like the patients with IGF overexpression, same mutation as in patients, and lo and behold, it didn't cause cancer. It caused a little bit of hyperplasia in these mice. So we made the mice with the colon cancer mutation as well, with the beta-catenin mutation in the Wnt pathway. They didn't get cancer. They got benign adenomas of the adrenal gland. But, as you might predict, I'm going to say, IGF ended up transforming the adenoma into a cancer, suggesting, again, that IGF may very well be an important initial mutation or second mutation in adrenal cancer, and important. In the germline, and this is a germline study here. So we started studying sporadic adrenal cancer when the genome study started coming out. And in our sporadic cancers now, not germline, just randomly occurring adrenal cancers, IGF-2 was always the most upregulated gene in any study ever done in almost every patient, like 90-fold excess, 100-fold excess. So again, some credence that it may be important. And so we went back to the basic side and started looking at IGF inhibitors that were being developed. There's about 25 of them that were developed in adrenal cancer cell lines. There you go. It blocked signaling. 1. 2, it blocked growth of adrenal cancer cells in culture. 3, we put adrenal cancer into mouse under the skin, and it blocked growth. So cool. So this started getting me excited in the possibility that what we were studying in the laboratory may have some potential translatability in the clinic. But Charles, I did not realize how hard this is to translate. So I begged and pleaded Big Pharma. There were 25 companies out there with inhibitors. I begged and pleaded for a little bit of their drug to do an investigator-initiated trial, meaning the company, for those who don't know, the company gives you money, university helps pay for the trial, or someone helps pay for it. I got no traction until finally I got a little bit of monoclonal antibody inhibitor from Pfizer, and this just shows you the few patients we treated. Above the bar means tumor growth, below the bar means tumor shrinkage. You'll see that in a small subset of patients, we had a biological effect, not necessarily clinically meaningful, but we saw tumor shrinkage with these end-stage patients, but short-lived, 10, 20% of patients, 4 months of a response. Did it again in a phase 2 trial, same thing, 50% tumor shrinkage, 4 months of response. But then we published these data, and a small company came to me and said, hey, we read all of your science. So students, publish your work. It's helpful. People read it. A small company came to me and said, hey, we want to do a phase 3 trial with you. We've read your data. We've got similar data. And they had a small molecule inhibitor to their IGF receptor. We did a placebo-controlled trial with our huge network now of 100 universities with over 130 adrenal cancer patients, placebo-controlled. And you'll see here that, again, we had responders. Small, only 20%, but this time, 80% tumor shrinkage with duration of response of up to 4 years, but only 20% of patients. So this shows you the kind of response, complete resolution of lung metastases, okay? But here's the challenge. It only worked in 20% of patients, but I just told you 100% of patients have the IGF mutation, IGF overexpression. So no one's going to be happy about that. Why? Because the return on investment wasn't there. It wasn't there for Big Pharma, 20% of 1,000 patients a year. It wasn't there for the government. Not enough lives saved. And we didn't have a biomarker. So we were done. We were dead. No return on investment. And there were 25 of these antibodies and small molecules for IGF receptor. They were all put on the shelf. The only success was adrenal cancer in this small subset. No other cancer they worked at all. So they're put on the shelf. And so what do we do next? Well, it so happens that it's about leverage again, because Michigan was put in charge of a few of the TCJ and the NCI projects to do deep sequencing of a few of the rare cancers. And adrenal cancer was one of them that we deep sequenced. And so years later, we do this study, and lo and behold, what we find is that adrenal cancer comes in 3 flavors: light blue, yellow, and dark blue. Dark blue is the most aggressive. Big genomic signatures, not important what they are for this discussion. But this study was $10 million, and it takes months to analyze the whole genomic data. And so what we did was, we did a component analysis just to ask the simple question: what are the main biological pathways engaged in adrenal cancer? Sporadic adrenal cancer, and very depressing to me, they were the same damn things we knew about their genetic syndromes: the Wnt pathway, the IGF pathway, and cell cycle control. So on one level, we learned nothing from this $10 million study. But the question was, is it possible that these 3 pathways underlie the 3 clusters, the 3 flavors: light blue, yellow, dark blue? So we're going to focus on IGF signaling. So what we did was, we did unsupervised analysis. We just took all the samples and said, what pathways are activated? Not just is IGF overexpressed, a whole activation signature unsupervised. And when we did that, they all had the same signature. IGF was activated in every cancer. So the unsupervised analysis gave us just one group. So that was consistent with knowing that IGF was upregulated in all 3 of our flavors. No surprise there. When we looked at the Wnt pathway, however, fascinating thing happened. In an unsupervised analysis, looking at Wnt activation, 3 groups emerged unsupervised. So that's interesting. But only 2 groups had a very profound Wnt activation signature. So interestingly, we got 3 groups, 2 of them had Wnt activation. I wonder if those are like the clusters, the 3 flavors. So we did what's called a network analysis, which is looking at the actual flavors themselves now and saying what transcription factors are active in those flavors. And lo and behold, the Wnt pathway transcription factors were only active in the yellow and dark blue. Again, consistent with these three groups being our 3 clusters and Wnt active in 2. Same thing with P53, we did the same thing. And this time, again, 3 groups emerged. But this time, only 1 had significant P53 signature, cell cycle activation. So is this happening in the 3 flavors? Well, the answer is yes. When we do network analysis, transcriptional activation of cell cycle only happened in that aggressive 3rd cluster. Why is that important? Well, here's why. Because what it means is we're able to detail the biology of these 3 flavors now by signaling pathways. And here it is. This is the slide. The money slide is they all have IGF dysregulation. 2 of them have additional Wnt activation, and a 3rd, only 1 of them has additional cell cycle control problem. Why is that important? These are targetable. And this is all about a talented student in the lab, Deepika Mohan, MD PhD student, who was able to take this multi-omic approach, which takes months, distill it to the biology, which takes weeks, and she distilled it down to an overnight PCR assay of these 3 flavors with 2 PCR steps. Okay? So the beauty of this is you can potentially use this overnight assay now to predict perhaps, I got to prove it to you, responders to IGF therapy, pre-selected, or Wnt or cell cycle inhibitors. I got to prove it to you. So the question for you all, well, what do? Do you publish your observations first or do you patent it first? Well, I had never done a patent before, but we were advised by colleagues like Dave here, who's a colleague of mine, to patent first. So that's the academic dilemma. Do you patent first or do you publish first? You want to publish first for your academic career, but we patented first. And we got 2 patents to define the 3 flavors with our overnight assay and to use those therapeutically to pre-select patients. Did those patents, and then she published her 7 papers as a grad student, which was pretty cool. But here's the story. Once we knew the biology here that could be defined by an overnight assay, as I show it again here, just a more complicated slide, but just the 3 flavors, we focus on IGF signaling and had the hypothesis, perhaps, that using this particular overnight assay, we could separate out these 3 flavors. What's the hypothesis? That the IGF-only patients may be the responders to IGF inhibitors. That light blue cluster that only had IGF signaling, maybe they were the responders. So went back to Astellas, I can say what the company was, went back to Astellas, begged and pleaded for the tumor samples that we had collected for the trial. And after about 2.5 years of negotiation, got the tumor samples. In the yellow bar, I just show you the responders. Not only were they the responders on the left, they're the long-lived ones who lived for 4 years. I know it's a small tail, long tail, but small number of patients. Nonetheless, there they are. Use our overnight assay in the lab. And after 2 years, as I said, to negotiate, we tested them. Every single responder was the IGF only. The non-responders always had a Wnt or P53 mutation in them. So that's cool. We had a diagnostic predictive assay then for a future trial. So that's the story about how I got involved. So with that as a backdrop, I went back to Astellas all excited like a kid and said, hey, you should do another industry-sponsored trial. We figured out who the responders are, and we have a CDX for that. CDX means patented companion diagnostic. So they said to me, well, what's our return on investment? Ah, shit. What's the revenue per year we're going to make? Back of the napkin was about 300 million a year. And they chuckled and said, well, no, thanks. That's kind of small for the market we're looking for. So somehow I had the audacity to ask them, would you license the drug to me? And they said, well, do you have a company? Sure. Yeah, I have a company. So that phone call ended. I started a company, Sling Therapeutics, with help from folks here, and then went back to them. And my goal was to then license my own technology for the diagnostic from Michigan, which was easy, pretty much, and then go to Astellas to get a license for Lincitinib. Well, that proved to be difficult. After another 2.5 years of negotiation, I got Astellas to do something, but it wasn't what you would want. Well, I had to settle for the following. They wanted $1 million down. I was in my basement basically during COVID and didn't have $1 million to give them, let alone 350. But I got them down to 350, which for them was a small amount, for me a lot. And they needed me to raise $10 million in 6 months, or else they're going to take the asset back. But they deferred the 350, didn't have to pay them that until I secured the 10 million. Sounds good, but COVID hit. Well, that was the problem. COVID hit. I could not raise any money because of COVID, and because the return on investment for VCs wasn't high enough either, Charles. It just wasn't enough money with this assay. And my 6 months ran out. I begged and pleaded for 3 extensions during COVID, but was finally given a drop dead date of April 21st, 2021. Hammer, if you're not done by then, we're taking this back. Just no more exceptions. So here's what happened. And this is about luck and the prepared mind. Louis Pasteur, in the field of observation, chance favors only the prepared mind. So my lab had been studying IGF in adrenal cancer. At the same time, at Michigan, it turns out, a colleague was studying IGF in another disease called thyroid eye disease, when the eyes bug out when you have thyroid disease. And they had proven it's important in thyroid eye disease. And it turns out, serendipitously, during COVID, another company called Horizon had just bought one of the failed IGF monoclonals. Remember, I said only ours worked in cancer. They chose one that had failed and was a monoclonal antibody. They bought it for $145 million untested. Remember, I got ours for $350,000. And they tested it in thyroid eye disease, and bam, it worked like gangbusters in 80% of patients. So that was an amazing story. They made billions of dollars after they got FDA approval in the first year. And then they published their work in the New England Journal of Medicine. Only then did my phone then ring off the hook. Okay. Lesson 4 or 5, be flexible. So my phone rings off the hook, and it's TPG, the 5th biggest private equity firm in the world, December 2020. I've got a few months to get a deal or I'm done. TPG calls me. They want to talk. I'm assuming it's about IGF stuff because they now know I have a drug. So I'm all excited. I tell them all about IGF signaling in adrenal cancer, but I have a couple of teaser slides about thyroid eye disease just in case. Well, you read it right. They had zero interest in adrenal cancer and were very interested in this disease I knew very little about called thyroid eye disease. So recall, I had about 2 more months to get a deal or I'm done. So my 6 month is running out, and with days to spare, in April 2021, TPG finally decides they're going to invest in my company with a $35 million Series A, with a valuation of $100 million, only because Horizon paid $145 for their drug, and they made 3 billion, and there was a return on investment now for them. So that's the lesson I learned, is you have to let go. You have to let go of your preconceived golden idea that you think is going to make your career. And I've studied adrenal cancer for 20 years. So by letting go, we founded a company. I gave away control. I hired a CEO. I'm not in charge anymore. I stayed on as chief medical officer for a year to get an FDA, an IND submitted, write a phase 2B trial, get 50 sites enrolling a trial, wrote 3 district protocols and 4 patents, and then realized I am way over my head. Lesson number 8, that they became a thyroid eye disease company. So lesson number 8 is the Peter Principle, which of course is all employees always rise to their level of incompetence. That's when you must walk away. That's what happens in so many industries, so many academics. So I had reached my Peter Principle, and I therefore said, I'm done. And I resigned from my own company because I was no longer effective. I wasn't useful anymore. It's a thyroid, they need an ophthalmologist. So I did that. I stayed on the board, founder and director, and we have top line data due in about 6 months with an IPO or a sale most likely pending on this phase 2B3 trial. With an $80 million Series B finance in the works with a new valuation of around $200 million. For a disease I never thought I was even interested in. So what did I do? I simply pulled my patents back, I pulled my patents from my company back to me, Gary Hammer, Michigan. So I still have my patents for adrenal cancer, that diagnostic. They weren't interested, pulled it back. And my hope is to do an investigator-initiated trial later. So we started out thinking about cancer, ended up doing thyroid eye disease. The question for the students in the room is, was this a success or is this a failure? Which was it? I've been studying adrenal cancer for 20 years, watching people die every day. Did I just sell out? Lessons learned. Aside from the ones I told you about, what are the other quick ones? Remember, we're all imposters. We're all doing things for the first time and never know what we're doing. I always feel like that. There's a first time for everything. You got to step up to the plate. It's a hard lift, but the magic always happens outside your comfort zone. Always. I want people to fear less and risk more. And this is how I feel. I'm a kid on a boogie board on a tsunami, always falling off. So you need a team. If you follow the science, you won't go wrong. Follow the science. Success is never a straight line, ever. And if you ground your questions in the best science, ground your goals in the best patient care, you'll do good. So I'll close with one more little video asking you the question, when you make your pitch, what's your pitch going to be? Listen to this, guys.
Being diagnosed with low adrenaline is devastating. No one ever warned me about adrenalitis, so I never saw it coming. There's times when you need adrenaline, and I didn't have it. That's when a friend who'd beaten adrenalitis recommended the all-new 245 horsepower V6 Toyota Tacoma with the TRD package. Knocked it right out. The truck's in.
The all-new Tacoma. Now that's moving you forward.
All right. So that's it. I thank everyone for listening to myself and Charles both, and I think we have a good 10 minutes for questions.
Okay, thanks, Gary. Thanks, Charles. Phil Messersmith, see your hand up?
No, I was just clapping. Thank you so much for both of those presentations.
Yeah, nice seeing you, Phil. Actually, Phil Messersmith here is actually a Catalyst awardee. He's at UC Berkeley, in fact, but I just wanted to mention that. Fantastic project of Phil's.
If there's no questions, we should hear about Phil's story.
Phil's story for the Catalyst project is really Miles Conrad's story, clinician at UCSF in interventional radiology. Anyway, it's a very early story, so I'm not sure there's really a whole lot to say there, but we're developing an early stage company for separation of tissues for thermal ablation of tumors.
Very cool. Nice to meet you.
So are there things that could be done. It seems like the main issue was there's just not a strong enough ROI. And there's reasons why there might not be a strong enough ROI. One could be too small number of patients affected. Another could be, larger number of patients, but not as devastating a disease. A third could be large number of patients, but it'll be really difficult to penetrate the market since there are other alternatives out there. Are there different strategies depending on what's leading to the same result of not a strong enough ROI?
Yeah, for sure. I think that, there's the need, the team, and the product. When the need is high, meaning and you have little competition, obviously then your strategy will be different because you may be first in class. What we argued early on, and then I'll let Charles chime in, is although IGF signaling is critical for adrenal cancer, it turned out that if you go to all the large databases, there are always rare 1, 2, 3% responders in all cancers. And there's a hypothesis out there that that if we can select those patients a priori, we didn't have a diagnostic for them, we think it may be overexpression of the IGF-1R, that you could do a bucket trial. But you have to prove that, perhaps other diseases or other similar cancers have the same defect that may be amenable, or other indications, which is how we went after it first and ultimately worked with Ted. Charles.
Yeah, the one thing I'll add is, what is interesting is that for rare diseases, for ultra rare diseases and just rare diseases, there is a return on investment. And so companies like BioMarin or Ultragenyx, Ionis, the RNA therapeutics company, they focus on rare diseases, and they're very, very successful because for reimbursement for payers, whether it's Medicare, Medicaid, or big insurance companies, they don't mind paying very, very large sums of money for individual patients' drugs because they don't have that many patients. And so it's sort of ironic, that for rare diseases and ultra rare diseases, there is a return on investment because, of course, there's the orphan drug considerations with the FDA. And so, and actually, Gary just drove this home. For many, maybe it's most, but for many at least, common sporadic diseases, there's also rare disease versions of it. And actually, Gary already mentioned that. And so not a game, an approach people take is target the rare disease, take advantage of orphan drug, maybe even the pediatric voucher, get the drug approved for the rare disease, paper in New England Journal, and physicians realize, oh my goodness, this might work on my patient that has the sporadic non-familial form. And oftentimes it works. Unfortunately, oftentimes it doesn't. So I do think that the government has to come in. I mean, there has to be incentive to develop drugs for these non, huge diseases like, cardiovascular disease, or I want to agree with what Gary said, and also just say that rare diseases are successfully treated.
If I'm not mistaken, a lot of companies now they're amassing portfolios of different rare, like BioMarin, of different rare diseases, becomes their portfolio, right?
Thank you, Charles and Gary, really for your just wonderful presentation. We have some junior faculty members who just at the start of their academic career, and then embarking on some of the translational projects. And, some of them are not at research institutions where they have this very elaborate network of support. What are advice that you can give them to just see when they run into challenges, and then how do they reach out and then get the help so they can overcome these problems to continue with their project development.
Charles?
Yeah, Yang, it's an unbelievably good point, because obviously if you're in Boston or Cambridge, Massachusetts, or the San Francisco Bay Area, there is this unique constellation of all the different components. And if you're living in a flyover state and there isn't that network, I completely see why you asked that question. I guess the one thing I'll say is that, one of the, I don't want to say it's an upside, but one of the things that COVID-19 pandemic yielded was, remote interactions, hybrid interactions. And so I think a lot of the things that, like the NCATS is sponsoring, or big accelerator programs make available, you can participate remotely, and there's Q&A and stuff. So I think if you are living in, I don't want to say a state, but if you're living in a state that's not Massachusetts or California or Michigan, it's just harder. But I think you could discover resources and have like a virtual community as opposed to an actual community. It's just a thought.
Yeah.
I'll just add to that. I'll just say one quick thing, that we have to try to train our trainees not to live in the silo and to be bold, fearless, and reach out. We can help them reach out, meaning we all as mentors reach out. Many people want to engage, whether they're clinicians or scientists engaged in space similar that can interdigitate with their science. People are interested, but it requires active effort for sure.
Yeah. So we have these 2 resource centers, right? We wanted to really be at service for everyone who wants to get involved in dental, oral, craniofacial tissue regeneration, and use this as one of the really important training materials for people. Maybe they can watch this recorded presentation and then follow up with questions. Really, this has been such a wonderful experience. Thank you again.
Okay, last call for questions. It's pushing 1 o'clock. Okay, great. I'll don't want to run over time again. Thanks to Gary and Charles for great presentations and opening our eyes to certainly some of the things to consider and some of the take-home messages in trying to evolve one's academic technologies out of their lab and ultimately into patients. So thanks again. I'll give you a virtual clap, and thanks for your participation.
Right.
Thanks everybody for attending.
Have a wonderful Thanksgiving next week, everyone. Thank you.
Likewise. Bye-bye.
All right.